Peter Houghton, who we have here today, received his PhD from the University of London. He moved to St. Jude's Research Hospital as a postdoc and chaired the Department of Molecular Pharmacology from '92 to 2009. He was the co-lead of the Solid Tumor Research Program from 1998, when he became director of the Childhood Cancer and Blood Diseases Institute at Nationwide Children's. Then from 2015 to 2021, he was the director at the Children's Cancer Research Institute at UT Health in San Antonio. Dr. Houghton has contributed over 400 publications focused on the development of preclinical models of childhood cancer and drug development for pediatric cancer, with his primary focus being on the biology and therapy of childhood sarcoma. And so with that, I'm going to turn it over to Dr. Houghton. Thank you for joining us today.
Great, thank you, Jay. So the title is Challenges to Generating Preclinical Data that Translates to Therapeutic Benefit. And it sounds easy, but it really isn't. So I'm going to sort of deal with different aspects of preclinical testing. In terms of conflict of interest, I'm a shareholder in Prolinx LLC, and currently receive funding from the various companies listed here, Solarius, Lantern, and OncoHeroes. And I'm a member of the NCI Pivot program, which has dealings with maybe 50 or 60 different drug companies. So this is on the web, so I won't go through it in terms of the summary of what I'm going to talk about. But the learning objectives are to understand the role of preclinical models in drug development for childhood cancer. And my focus has been childhood cancer exclusively for the last 45 years. And understanding the major limitations of preclinical models, because I think if you understand the limitations, then you can build better models, or you can build better experimental designs that more accurately utilize the models that are available. And understanding how alternative experimental designs for preclinical testing may lead to more accurate translation to clinical trials. And we'll try and get through all of this in the next hour or so. So what is a patient-derived xenograft? Well, for those of you who have been out of circulation for 20 or 30 years, it's not a new idea. Patient-derived xenografts, wherein a human tumor is transplanted directly into immune-deficient rodents, have become the predominant preclinical models for drug discovery and drug development. However, all models have deficiencies that limit their value in translating preclinical results to clinical application. And we're going to deal with some of those limitations today. So we have to set where we're going with models in terms of the current legislative imperatives. And that is the RACE Act, the Research to Accelerate Cures and Equity for Children. And this requires the FDA to develop a list of molecular targets and molecular targets of new drugs and biologics that are determined to be substantially relevant to the growth and progression of pediatric cancer. And if so, this may trigger a requirement for pediatric investigations. Again, the Act has the intent of engaging pharma in pediatric drug testing at an early stage in drug development. So the Act would necessitate having the ability to test novel targeted agents against models with appropriate molecular characteristics. And this will require establishing PDX models of specific diseases having the appropriate molecular characteristics. So why do preclinical models fail to accurately translate in cancer drug development? And one has to set this in the concept that 95% of drugs that enter into clinical trials fail. Okay, so what is it that we are not doing at a preclinical level that allows drugs to go forward that ultimately fail in the clinical situation? And I think we can divide the failures into basically 5 different categories. And I'm really just going to deal with the top 3 during this talk. But there are differences in species tolerance to drugs, the ability of models to recapitulate human molecular and genetic heterogeneity. Criteria for defining activity is less stringent in preclinical trials compared to clinical trials. And unpredicted toxicities that occur in patients, but not at preclinical toxicity models. And then the 5th, and this is becoming more important, is that most preclinical models, and PDXs are a classic example, do not comprise a functional immune system, and responses to therapeutics focus essentially on a tumor-intrinsic drug sensitivity. So I'm going to go through the first 2 of these in some detail and mention the 3rd. So differences in species tolerance to drugs has not been considered critical for accurate translation. So what do I mean by that? It means that we've essentially said if a tumor responds to a drug at the MTD in mice, that tumor type should respond in patients. And that's obviously very naive, but the reality is the great majority of papers that are published that suggest a drug could be useful in the clinic have no pharmacokinetic data to back that statement up. So let me give you an example of how we can go horribly wrong in preclinical testing. So this is the activity of an acylfulvene derivative, MGI-114, that's in development at the turn of the century. And if we just focus where the arrow is on BT27, which is a PNET, this is 11 brain tumor PDX models. And if we look at the dose reduction, so 4.6 milligrams per kilogram per dose is around the MTD for the PDX models. And we reduce the dose to 3. So one sees that at 4.6 and 3 milligrams per kilogram, one gets complete maintained responses. So the tumors are very responsive. But as you go to 2 milligrams per kilogram, you get stable disease. And at 1.3 milligrams per kilogram, you have progressive disease. And at 1.3 milligrams per kilogram, all of the models tested are progressive disease. So this looks like a very active agent, certainly at the MTD in the mice. So how does the exposure at the MTD in the mouse compare with the exposure in humans? This is work from Gail Eckhart. In the yellow bars show the AUC, or area under the concentration curve, for patients as you increase the dose from 5 to 15 milligrams per kilogram. And it's shown here, the AUC in humans is very low relative to the AUC in mice at the MTD, which is tens of fold higher. But even as you reduce the dose, where you have 0 out of 11 responses, as shown in the previous slide, we're still way higher in terms of exposure in the mouse than we can achieve at the MTD in a human. So does this metric actually hold for other tumors? So if we take the drug exposure at the mouse MTD and divide that by the drug exposure at the human MTD, we see that drugs that are clinically not active, which is the top 6 here, that we have ratios of 15 to 20 fold. So what this means is that the MTD in the mouse, you have 20 times the exposure that you can achieve in the human. And yet, if you look at the effective range in the mouse, reducing from the maximum tolerated dose, it tends to be in the order of 2 to 3. So in other words, even when you're losing activity at a third of the MTD, you're still probably 5-fold, at least 5-fold higher than in terms of AUC than you can achieve in the clinic. Carl Zellison is a perhaps the example where the mouse is about 80-fold more tolerant than the human, yet the effective dose range is less than 2. So even when you lose activity, you can still be 40-fold above the level of drug exposure achieved in a human. And Solofina MGI-114 is the one I just showed you. The PPTC recommended forwarding alisertib, an Aurora A kinase inhibitor, for clinical trial. But in reality, the exposure in mice at the MTD is around 8-fold greater than that in children. And the effective dose reduction range, that is the dose relative to the MTD where we lose activity, is around 3. So again, it's very unlikely that this drug will be active in a clinical setting. Similarly, the prodrug, PR104, which is activated by AKR1C3, again, the exposure in mice is greater than 4-fold that you can achieve in humans, but the effective dose range is less than 3 from the MTD in mice. So again, there's little anticipation that this drug will be active in solid tumors, but on the other hand, in T-cell ALL, which has a high level of AKR1C3, there is potentially some activity. If we look at clinically active agents, melphalan, topotecan, and irinotecan, we see that at least for melphalan and topotecan, the exposures are very similar between mouse and human. The dose reduction before you lose activity in the case of topotecan is 10, and that for melphalan 2 to 3. So you would expect these drugs to be effective. And similarly with irinotecan, which is very well tolerated in mice, has a very wide range of activity. So again, one would expect that those exposures that can be achieved in humans, that one would see responses in the appropriate models or human cancers. So I'm going to move on to the second part, and that is tumor models do not recapitulate human cancer at a molecular or genetic level and encompass clinical heterogeneity. And I'm going to divide this up into 2 parts. Retention of molecular genetic characteristics, and then encompassing clinical heterogeneity. So this is work that's recently been started by Siyuan Zheng at the Greehey Children's Cancer Research Institute with assistance of Funan He, a postdoc. And we've developed about 84 new PDX models. And we've started to characterize these at a genomics level. And we've made specifically an attempt to develop PDX models from minority underserved patients. So a lot of these are from Hispanic patients who tend to do somewhat worse in terms of leukemia responses and in terms of their responses to certain drugs. So what we've done here is to look at germline, patient-derived xenograft, and patient tumors with respect to RNA-seq. And we've done germline patient tumor PDX for whole exome sequencing and genomic sequencing. And the data set consists of solid tumors most commonly seen in children, and including germ cell tumors, hepatoblastomas, osteosarcomas, various kidney cancers, etc. And we have a similar number of new PDXs developed from leukemia patients, of which 65% of those were derived from Hispanic children. But what I'm going to show here, so what does that mean? So PDX models largely recapitulate the molecular heterogeneity of the patient tumor, around 70%. There are exceptions where the PDX represents a subclone of the patient tumor. And there are examples where there is a reduction in mutational complexity in the PDX, where a precursor-like clone becomes the predominant clone. I think this data, and obviously there are other data to support this, is that it cautions against this concept of co-clinical trials. In a co-clinical trial, you heterograft a tumor into a mouse, and then you treat the mouse with the intent that when a patient relapses on therapy, you can identify drugs that are effective or could be effective in that patient case. So the expectation or the anticipation is that the PDX model will recapitulate the sensitivity of the donor patient and hence predict drug sensitivity. But I think these two examples where the PDX represents a subclone of the patient tumor, or where there's a reduction in clonal heterogeneity in the model, clearly raise the question of whether such co-clinical trials are meaningful, particularly in the context of pediatric cancers. So, encompassing clinical heterogeneity. We know that medulloblastoma, rhabdomyosarcoma, neuroblastoma, and ALL, obviously, represent different molecular entities. In the case of medulloblastoma, certainly by expression profiling, you can group these into at least 4 groups, and maybe each of those groups has a subgroup. And when one considers that there's only about 220 patients a year in the US, you start dividing the pie up, it becomes progressively very difficult to assess the response of molecularly targeted drugs that should be active in different subtypes. And I think it's here that the models become really quite valuable. The rarer the molecular subtype of tumor, the more valuable models become as a way of assessing whether a drug should proceed further into clinical trial. In the case of leukemias, there's 17 different subtypes that are represented here. So even with greater than 3,000 patients a year, there are entities which are very rare, even in leukemias, where models would be quite valuable to develop. So we are part of the PIVOT program, which is the successor to the Pediatric Preclinical Testing Program and Consortium, which is involved in testing new drugs against pediatric cancers. And I think within the consortium, we have around 700 patient-derived xenografts and cell-derived xenograft models of childhood cancer. These include neuroblastoma, various flavors of sarcoma, brain tumors, kidney tumors, hepatoblastoma, and leukemias. So I think the PIVOT program is well situated to facilitate testing that will be required by the RACE Act. So what does the RACE Act mean for preclinical testing? Well, it means for pharma and academic centers, the RACE Act will potentially require preclinical data to support or refuse the activity of a compound being advanced to testing in children. This may require development and characterization of hundreds of PDX models to simulate the genetic and epigenetic characteristics required. But we already know that having a particular aberration, say a BRAF V600E mutant, may or may not result in sensitivity to a given agent. For example, whereas V600E mutant melanomas are quite responsive to BRAF targeted agents or MEK targeted agents, the same mutation in in colorectal cancer does not necessarily confer sensitivity. And similarly, in a pediatric context, the responses to MEK inhibitors in low-grade glioma are somewhat different from those that we get in glioblastoma. So response is context specific. So we have to develop models of specific tumor types that have the relevant molecular characteristics for drug testing. So potentially there are 200 new agents that may require preclinical testing each year. Oops, sorry. So this becomes a logistical challenge, one could say a nightmare, but let's see how we can do it. So I think we have to incorporate molecular diversity required by the RACE Act into testing procedures. And for that, testing procedures have to evolve. So let's go and look at conventional testing. So this is with group sizes of 8 to 10 mice per treatment group. And that's traditionally been used to assess the statistical significance of a therapeutic response. The consequence is that few models are tested. So in the PPTC and pivot programs, one would necessarily test 6 neuroblastomas, for example, or 6 rhabdomyosarcomas. And so it probably precludes incorporation of the molecular heterogeneity that is required to fully recapitulate the human disease. I think the second point is we've been driven by this ridiculous idea that a p-value of 0.05 is actually meaningful. Statistical significance does not mean biologically meaningful anti-tumor activity. How many times have you read papers often in high impact factor journals, which figure 6 is the in vivo validation of a target. And what you see is about a 20 or a 30% inhibition of tumor growth. But the 30% inhibition of tumor growth is tumor progression. And in clinical trials, we're looking for stability of disease or tumor regressions. We're not looking for a 30% decrease in tumor growth rate, because firstly, we can't measure it. And secondly, this is progressive disease, which means the drug is not being sufficiently active. So I think preclinical testing should identify agents that have potentially meaningful biological activity. And by meaningful biological activity, I mean the ability to cause tumors to shrink or disappear completely. So I think alternative experimental designs can enable inclusion of molecular heterogeneity. Retrospective analysis of over 2,100 tumor drug experiments from the PPTP using 8 to 10 mice per treatment group was undertaken and was published in 2016. And basically what we did was to take a very large data set of some 67 drugs, 83 tumor models, and 2,100 studies, and simply asked, would we obtain the same result if we use a single mouse rather than the 8 to 10 mice per treatment group? So the way we did this, and this is just to show the approach, is that let's suppose the group response in our conventional testing was stable disease. And the single mouse selected at random from that group was also stable disease. That would be scored as 0. If the random mouse had been a progressive disease, 2, then we would score it as a -1. If the group response was a stable disease, but our random mouse was a partial response, we would say it's overpredicted one response category. So the worst we can do is we can underpredict by a numeric value of 5. So in this case, our mouse is progressive disease, whereas the group response is maintained complete response, or vice versa, overpredict by a value of 5. And the results are shown here. So if you take this 2,100 different experiments and you say, let me take one mouse out of that group, did it give me the correct answer? And the answer is, in 76% of the time, it was exactly right. And if you take a little bit of wiggle room, such as one response criteria of under predicting by one response category or over predicting, you came up with 95%, which is not bad for a preclinical screen. Okay. So you could argue from this data set that we looked at a lot of inactive drugs, and believe me, we looked at a lot of inactive drugs during that period of 10 years of testing. But so you can look at the same sort of type of analysis in a different way. You can say, what is the objective response rate for the models to a particular drug from the group objective response versus, again, the single mouse that you select out at random from that group? So, for example, let's suppose we tested 50 models, and in 25 of those models, there was at least a partial response. We would say that the objective response rate was 50%. So again, looking at the group objective response rate versus the single mouse response rate, the red lines are the data that was generated. So you can see that the group response and the single mouse response are similar for drugs that have virtually no activity, so less than 10% objective response rate in the models, to those that, in this case, one drug that gives 100% response rate in 5 models in leukemias. Okay, so anyway, in between, from inactive to very active, the relationship between the single mouse and the group response is remarkably good, with perhaps one exception, and that's shown at the bottom here, where the single mouse predicted a 50% response rate and the group response was 0. And this is tested only in 2 models. So it's an outlier. The blue lines, blue dots rather, are 1,000 simulations of the data done by the statisticians on the paper. And as you can see, the simulations and the actual observed data are remarkably similar. So we tested this prospectively, both in leukemia models and in solid tumor models. And I'll show you a couple of examples here. So this is from data from Richard Locke at the Children's Cancer Institute in Australia. And Richard is part of the PIVOT program. And he ran topotecan against 80 models of acute lymphocytic leukemia, and they're shown in the top panel. And this was testing topotecan, which is a fairly active drug in leukemia. And you can see the responses of decreases in percentage of human CD45 cells from baseline. So the drug's very active, with a few examples of tumors such as these MLs that really don't respond to topotecan therapy. So the idea would be, if we could create enough models, can we actually start teasing out tumors that respond versus those that don't respond, and really start to understand biomarkers that might be predictive of response? But in this particular case, what we were looking for is similarity between the single mouse data, which is this data at the top, and the same tumor models run using a conventional design of eight mice per group. And those 7 tumors are marked with the white arrows at the top. And the table at the bottom, shown here, shows those tumor models, and it shows the response to conventional testing, maintain complete response in red. As you can see, ALL2 is the same. In the case of ALL3, single mouse testing overpredicts by 1 response category. ALL4, both progressive disease. We overpredict in ALSF by 1 response category, etc., and the rest are the same. The difference here is that the conventional experiment takes 128 mice, and the single mouse experiment takes 7 mice, and essentially you get the same result. Okay. If you were to take this whole experiment with 80 different ALL models, it would take 80 mice versus 1,270 mice. So in terms of resource conservation, the single mouse experiment seems to give us the same data as conventional testing. It costs a fraction of the cost because we use far less mice, but potentially it allows us to do more experiments where we really can look at novel agents against a wide spectrum of tumors of the same type, and perhaps start to tease out biomarkers in response and non-response. But in some respects, ALL is not a great example because one would like to sort of generate 50 of each of these models or 70 of each of these models to be able to do the same sort of analysis in leukemia subtypes. We've done the same sort of prospective analysis of single mouse versus conventional testing with solid tumors. This shows a single example of trastuzumab deruxtecan, and it shows on the left individual tumors. So each line represents a different tumor model to treatment with this antibody drug conjugate. And you can see there's a range of responses from really progressive disease to regressions and complete maintained responses. So this is clearly an active drug. And again, if we do the comparison between medium response to conventional testing, in this case, 10 mice per group, we see that BT29, the data is the same. The single mouse overpredicts for 1 response category in a rhabdoid tumor in terms of KT10, and the other tumors were essentially identical results. So again, instead of using 50 mice, we can use 5 mice to do the same, or to get the same data. The question is, do we really need statistical analysis when you see tumor regressions? I think you need statistical analysis to get things published, and when the drug really doesn't do very much. But when you have a drug that causes complete regressions in tumors, then I think we're less likely to dwell on the value of the statistical analysis. So what that does allow us to do is to divide the tumor models up into poor responders, intermediate responders, and exceptional responders. And if we have the adequate numbers, in the case of DS7300A, we had 34 tumor models. We now routinely run up to 60 tumor models in single mouse testing, which incorporates both the heterogeneity of tumor models, because sometimes you don't know what a drug is going to be active in, but also a substantial number of models of the same type that incorporate some of the molecular heterogeneity that one would find in a clinical situation. So I think the single mouse testing design adds power to biomarker identification. So I'm going to sort of switch gears a little bit now and say, can we actually recapitulate clinical trials at a preclinical level? So if we use single mouse testing, we can certainly incorporate a lot more tumors than we can with conventional testing within the same resource constraints. So this is results from the 0431 clinical trial with high-risk rhabdomyosarcomas that was published about 5 years ago, from Brenda Weigel. And it shows the outcome for patients who had less than 2 Oberlin risk factors in the yellow line, and those that had 2 or greater Oberlin risk factors. And the rather sobering point of the blue line is that you can superimpose the survival with all of the IRS studies that were done in the 70s through 90s. So we really have made very little progress in terms of improving therapy for patients with high-risk rhabdomyosarcoma. So can we use single mouse testing to simulate a clinical trial and understand intrinsic and acquired resistance? So here's a couple of Kaplan-Meier curve. The blue line on the left is the time to event for each of these tumor models that does not receive treatment. So you can see that they progress fairly quickly. And the orange line shows the response to a single cycle of VAC therapy. So we're not trying to simulate 52 weeks of treatment that you get in the clinical trial. We're mainly looking at the intrinsic sensitivity of each of some 34 rhabdomyosarcoma models. And you can see that from RH87, which fails very rapidly, through to the tumors at the end, RH12, 28, and so on, that are maintain complete responses over 100 days after a single course, that you have a spectrum of tumors that have different intrinsic sensitivities to vincristine, actinomycin B, cyclophosphamide PAC therapy. And again, it's a model where you can start to mine the data to say, are there signatures of those tumors that are responsive, and are there signatures of tumors that are non-responsive to PAC therapy? Similarly, we can do vincristine irinotecan, which is another component of the ARST 0431 protocol. And again, you can see that the therapy, a single cycle of VI therapy, pushes the progression-free survival to the right very considerably. And you can ask the question, are the tumors that are responsive to VAC therapy the same tumors that respond to VI therapy, or is there cross resistance? And you can see examples where there's tumors that are responsive to neither drug, such as drug combination, such as RH87, and you can see those that are responsive to, very responsive to both drugs, such as RH12, RH28, RH36. So again, it's a useful approach to try and simulate clinical combination therapy in a preclinical level. What it allows you to do is to take tumors where you know there's resistance to VAC or resistance to VI, and really focus on those, because those tumors that are resistant to those drugs would be largely the patients you're likely to get in a phase 2 trial. So what you would really want to do is to say, I have a new drug that is as equally active in these VAC resistant or VI resistant tumors as they would be in sensitive tumors. So I think it gives us an opportunity to rethink how we do preclinical testing. That is tumor context specific, but also you could do exactly the same experiment with respect to a molecular characteristic. That is to look at different tumors that have BRAF mutations, and then ask the question about sensitivity in that context. So I'll go through as a summary each of the things that I've discussed. So differences in species tolerance to drugs is actually absolutely critical for accurate translation. In many cases, when we get a drug through Pivot, there is some pharmacokinetic data available from adult trials. And although PK may differ between adults and children, it's at least a guideline to say, can we recapitulate the exposures in mice that are relevant to adult pharmacokinetics or exposures, and that includes both the Cmax as well as the rate of clearance. So exposure, whether it becomes important, whether the drug activity is driven by the Cmax or just by the exposure or time above a trough level. So these things can be factored into preclinical testing. They tend not to be. It's often very difficult to get pharmacokinetic data to allow us in Pivot, for example, to dose mice that in a way that would be relevant to clinical exposures. And I think we really have to push that further. In terms of testing, we tend to test a single drug dose or schedule. And I think with single mouse testing, we can probably, within the same financial restraints, look at multiple dose levels and potentially different scheduling that's relevant to clinical dosing and administration of drugs. And we need to take time to do that before a drug is advanced to potential clinical application in kids. And I think over the last 20 years, we perhaps have rushed some drugs to be considered for clinical testing that should not have gone that quickly. So tumor models don't recapitulate human cancer. This is something that Bob Weinberg back in 2004 raised at an AACR conference. And I think it's something we had to consider. I think with all the evidence suggests that the great majority of pediatric PDXs maintain most of the characteristics that we want to see, and that we can construct models that have specific genetic lesions that may be the targets of novel drugs. So I think at preclinical level, we've got a large number of models where we can draw from that would be relevant to testing most drugs. Do we encompass clinical heterogeneity? I think the issue there is, yes, we can develop models that encompass clinical heterogeneity, but we need to really rethink how we do this in terms of preclinical testing that allow us to incorporate that clinical heterogeneity into preclinical trials. And Finally, the criteria for defining activity is less stringent in preclinical trials compared to clinical trials, and we dealt with this sort of slightly during this talk, but the reality is we're looking for drugs that cause tumors to go away, to disappear, and hopefully, for considerable periods of time in mice, for those drugs to be really considered as being valuable for moving forward into pediatric clinical trials. I haven't dealt with unpredicted toxicities, but I think good examples of that would be the MDM2 inhibitors, where in humans they cause thrombocytopenia, but in non-human models that toxicity was not seen. And in part, this is because this class of drug was developed against recombinant human proteins, and the drugs have much greater affinity for those human proteins than they do for mouse or rat or dog proteins. So the preclinical models no longer become useful toxicity models. And as I alluded to earlier, the PDX models don't have a functional immune system, and responses to drugs focus on tumor intrinsic drug sensitivity, which may only be part of the question. As we move forward developing humanized mice, we'll see how valuable these models are in the context of immune oncology development. So with that, I'd like to thank all those who are part of this program, in particular Roshan Kamasheva, who co-leads the Pivot program at Grihi, and particularly Xion Zhang and Funan He, who are conducting the bioinformatics analysis of new PDX models that we've developed under a secret grant. So with that, thank you, and I'll try and take questions.